A system is a group of interacting and interdependent elements. For example, a system could be a group of planets and stars connected by their attractive forces, or different components assembled together in a computer. In order to understand how a system works, it is not sufficient for us to only study the individual parts, but rather we need to study how they work together. For instance, if we take a mobile phone apart and identify all its pieces, we still wouldn't understand how the phone makes calls or sends messages. This is because a phone, or any other system, depends entirely on how its parts work together. The human body is also a system, a highly complex one, and many diseases appear when not one but several parts of the body and the connections between them don't function correctly. Until very recently, we could only examine these parts of the body in isolation, and the study of complex diseases, in which several genes and proteins are involved, represented a major challenge. However, the technological advances of the last few years now allow us to generate huge amounts of data and to analyze this information so that we can better understand how our body works. This new approach to the study of human diseases is known as systems medicine. Using systems medicine, we can build mathematical models to study the connections between different genes and proteins that work together, and then we can identify which combinations of these are responsible for a disease. In this way, we get a clearer picture of complex diseases, allowing us to improve their diagnosis and treatment. The European project CombiMS is coordinated by Bionure, a Spanish company that is pioneering the use of systems medicine to find new therapeutic drugs. This project is a collaborative effort of leading European clinical centres and experts in systems medicine, applying this new approach to study multiple sclerosis, a classic complex disease. In multiple sclerosis, changes occur in the immune system that cause it to attack the body's own nerve cells, producing severe physical problems. At present, there are no treatments available that can halt or reverse the progression of this disease.
In CombiMS, we have studied immune cells from patients with multiple sclerosis. We have collected cells and we have analyzed the phosphoproteome, which mediate the signaling between the signals coming to the surface of the cells to the DNA. Now we have generated a big amount of data, and with data we have applied to computational and mathematical models we have developed in order to make sense of this information. Now we have generated models how the disease and how the cells behave during this inflammatory process, and we have identified fingerprints that can be used as biomarker of the disease and can be used as signature of the damage process of the brain.
These signatures should help increase the accuracy of diagnosis and aid in predicting the progression of the disease, as well as helping to better understand the action of different drugs. In turn, this will allow patients to receive a much more personalized treatment.
In order to treat chronic diseases like multiple sclerosis, the best approach is using combination therapy, meaning different combinations of drugs. But in order to do that, we need to understand which are the best combinations in terms of better efficacy, but more importantly, preventing side effects. With the models we have developed in CombiMS, we plan to simulate the combination of drugs and predict which combinations are going to be more effective, but especially which one we are going to prevent to have adverse events. And this is going to be on the benefit of people with multiple sclerosis.
Ultimately, CombiMS has set out to find a better way to study not just multiple sclerosis, but also other complex pathologies. By bringing together new technologies, biology and medicine, it is expected we will find new and effective solutions to understand and treat human diseases.